Welcome back to this next video in which we are discussing tuberculosis and anti-tuberculosis drugs. Okay, so we're currently in the process of discussing the adaptive immune response, which is going to be a T helper 1 mediated adaptive immune response against Mycobacterium tuberculosis. So remember, so far what we've seen is the antigen presenting cell has successfully, in this case, phagocytosed a Mycobacterium tuberculosis bacterial cell, and it's now presented the fragments of antigens from Mycobacterium tuberculosis on its cell surface in the context of MHC class II protein complexes. It has then gone to a mediastinal lymph node via lymphatic vessels, and it is now attempting to activate CD4 positive or helper T cells that are currently in the naive state. Now remember, it can only activate CD4 positive T cells because CD8 positive T cells can only be activated by peptide fragments on MHC class I. So it needs to find CD4 positive T cells which have T cell receptors on their surface which can recognize one of the peptide fragments on the surface of the antigen presenting cell in the context of MHC class II. And remember, there are a huge number of different types of helper T cells which will have different T cell receptors. So a useful concept is the concept of a T cell clone. So the concept of a T cell clone means all of the T cells with an identical design of T cell receptor. So the antigen presenting cell needs to find T cells of T cell clones which have T cell receptors which can recognize and bind to one of these MHC class II peptide fragment complexes that the antigen presenting cell has on its surface. So it will have a job finding T cells of that specification. So we are now following the story of one of these, however. So we're assuming that our antigen presenting cell has managed to find a member of a T cell clone where the T cell receptor can recognize one of these peptide fragments when it's presented in the context of MHC class II. And this is what is now happening. So here's the MHC class II with the peptide fragment there. It's bound to the T cell receptor here, and the co-receptor on the surface of this CD4 positive T cell is CD4, of course, and that's also bound to the MHC class II peptide complex here. Once you've formed that, the T cell receptor and the CD4, they deliver signal 1 to the naive CD4 positive T cell. In addition, you have to receive signal 2 if you're going to proceed with the activation of this naive CD4 positive T cell. Otherwise you go into an anergic state, and we discussed at length in the previous video why that is. So the antigen presenting cell, because it's come into contact with Mycobacterium tuberculosis, which is most certainly a pathogen, it will have B7 molecules expressed on its surface, which can be both B7.1 and B7.2 molecules. They can both do the same thing. They can bind to CD28 receptors on the surface of the naive CD4 positive T cell, and when that occurs, CD28 will deliver signal 2 to the naive CD4 positive T cell. Signal 1 and signal 2 will then result in this naive CD4 positive T cell expressing two crucial molecules, which are interleukin 2 and the interleukin 2 receptor alpha. The interleukin 2 receptor alpha protein will go to the cell membrane, where it will join up with interleukin 2 receptor beta and interleukin 2 receptor gamma proteins, which are already expressed by the naive CD4 positive T cell, to produce a full interleukin 2 receptor, which is what we've got shown here. So this now is a full interleukin 2 receptor. Interleukin 2 will be secreted from the cell, and then autocrine signaling will occur where the interleukin 2 activates the interleukin 2 receptor on the same cell that secreted it, and that then delivers signal 3 to this naive CD4 positive T cell, and we want to follow the story from there. Now I'll remind you that there might be multiple antigen presenting cells bringing the peptide fragments from antigens of Mycobacterium tuberculosis cells to lymph nodes, and they might be activating different T cell clones, because remember, The Mycobacterium tuberculosis, it has many antigens. All of these antigens have multiple peptide fragments, so we can launch adaptive immune responses of this T helper 1 mediated type, where the T cells are going to be directed against all of these different peptide fragments, and they'll all be being launched concurrently. So you won't just be launching one of these. We are following the story for one of these for simplicity, but remember that you'll actually be launching multiple of these concurrently. So now let's go back to just discussing this for a single peptide fragment. So what is this specific T cell now going to actually do? So upon receiving signal 3, you might be able to guess what's going to happen. Our naive CD4 positive T cell is going to start dividing and dividing and dividing and dividing. So I'll draw this here. So we go from having one cell to having a whole bunch of cells. So we actually can call this T cell clonal expansion, because all of these progeny T cells that you're producing here, they will all have the identical design of T cell receptor. They'll all be covered in this exact same T cell receptor here, i.e. they'll be members of the same T cell clone as the original T cell which we activated. So we've actually hugely expanded the population of that T cell clone. So here we go. We are expanding our T cell clone. So this process is known as clonal expansion. Okay, what can now happen is these CD4 positive T cells that are still currently in the naive state, they're not in the effector state yet, they can now start to differentiate into cells which are actually going to do something. And the two different types of cells that they can differentiate into are memory T cells and also effector T cells. So what we're now going to start getting is differentiation, and some of these T cells that we produced here will become memory cells, but most of them will become effector CD4 positive T cells or effector helper T cells. So let me just draw this here. So I've drawn out 4 of these cells that we produced. In reality, you'll produce a much bigger population than just 4 of them. So to illustrate the point that most of them will become effector helper T cells or effector CD4 positive T cells, I will have 3 of them becoming the effector T cells, and then I'll just have 1 of them becoming a memory T cell. So here, this will then be a memory T cell. So we're going to get differentiation here. So let me firstly just discuss the memory T cells and what they're going to be doing, and then I'll come on to discuss the effector T cells. So this is a memory CD4 positive, or you could call it a helper T cell. Okay, so the memory CD4 positive T cells, they actually have a very similar function to the naive CD4 positive T cells. And let me just give you the motivation for why you would want to produce memory CD4 positive T cells. The idea is this. Let's say the effector CD4 positive T cells were capable of clearing the pathogen. Okay, in tuberculosis they're not going to be capable of clearing the pathogenic infection, but let's say for simplicity that they were capable of clearing the pathogenic infection. What if you were then reinfected? You see, the problem with effector helper T cells or effector CD4 positive T cells is they have a very short lifespan. Effector CD4 positive T cells, these green ones here, they generally only live for about a week. Compare that to the lifespan of the naive CD4 positive T cells, which have a lifespan of up to a year, from months to years is the lifespan of the naive CD4 positive T cells. Okay, so the problem is, if we were to convert our naive CD4 positive T cell into solely a huge great population of effector CD4 positive T cells, then all of those effector CD4 positive T cells, they might be able to clear the pathogenic infection, but after a week they're going to start being removed from the body, they're going to die, and they'll be removed from the bloodstream, and then that clone of T cells, the T cells with that T cell receptor, might have completely died out from the body. Okay? That would cause a problem. You don't want that to happen. So you produce these memory CD4 positive T cells to replace the naive CD4 positive T cells, and they're there to be activated if you were to be reinfected by the pathogen. So memory CD4 positive T cells, they have a very long lifetime. They can live for the entire lifetime of the organism, and you'll have them in a much greater population than the initial naive CD4 positive T cells. So for a single T cell clone, you might have only had one, let's say, naive CD4 positive T cell with that specific design of T cell receptor. After you've activated it and produced lots of memory CD4 positive T cells, you'll produce loads of these, so you'll have a much greater population of those memory CD4 positive T cells of that T cell clone than the initial naive CD4 positive T cells. And these are responsible for why a secondary adaptive immune response is much quicker and much larger than a primary adaptive immune response. So when you're first exposed to a pathogen for the first time, you initiate primary adaptive immune responses, and those take a long time to initiate, and the reason is that the T cell clones and also the B cell clones, but we won't dwell on that because we haven't really discussed B cells, but the T cell clones are small. The population of cells of each T cell clone are very small, and therefore it takes a long time for the antigen presenting cells to actually correctly find T cells of the correct T cell clones to activate. Whereas this secondary adaptive immune response, which is where you're exposed to the pathogen for the second time around, that's much quicker because of the fact that the T cell clones that you need to activate, and also the B cell clones, but as I say, we're not dwelling on that. The T cell clones that you need to activate, they have a much greater population. You've got a lot of these memory CD4 positive T cells, so it's going to take much less time for the antigen presenting cell to actually find one of these memory CD4 positive T cells to activate, and therefore you'll get the secondary adaptive immune response occurring much quicker. And of course this is the basis of why we vaccinate people. Okay, right. So, you produce those memory CD4 positive T cells which replace the naive CD4 positive T cells, and they can be reactivated by other antigen presenting cells and will then do the same thing as the naive CD4 positive T cells. So once they've been activated in this exact same way, they'll then divide and divide and divide, and some of the cells will differentiate into effector CD4 positive T cells, and some will then differentiate into memory CD4 positive T cells to maintain the memory cell population. Okay, so let's now go on to the effector effector CD4 positive T cells, also known as helper T cells. Okay, so there are different types of effector helper T cells. So for instance, T helper 1, T helper 2, T helper 17, T follicular helper, the list goes on and on. There are loads of different types of effector CD4 positive or helper T cells. In the adaptive immune response to tuberculosis, the effector helper T cells that you're going to produce are going to be of the T helper 1 type. And I now want to explain the reason for them being the T helper 1 type. So when these cells are differentiating into effector helper T cells, the type of effector helper T cell that they're going to differentiate into is determined by the presence of signaling molecules from the site of infection. And in particular, there is going to be a signaling molecule present in tuberculosis infections known as Interleukin 12, and this acts on these differentiating cells and makes them differentiate into the T helper 1 type of effector helper T cell. Okay, so Interleukin 12 acting on these differentiating CD4 positive T cells promotes them becoming T helper 1 type effector helper T cells rather than T helper 2 type or T helper 17 or T follicular helper or T regulatory. Okay? Now, the question then of course becomes, why do you have Interleukin 12 present in the lymph nodes where of course all of this is happening? And I should have stressed that actually. these T cells will still be in the lymph node where they were activating. So this population will have expanded in the lymph node. The differentiation will be occurring in the lymph node. So why do you have this presence of these interleukin-12 signaling molecules in the lymph node in a tuberculosis infection? Well, the reason is that the phagocytes, so macrophages for instance, at the site of infection that are coming into contact with the Mycobacterium tuberculosis cells, they are activated to produce interleukin-12. So if I go back to a previous picture here, so these cells here, the neutrophils and the macrophages, all of these immune cells that are coming into contact with Mycobacterium tuberculosis, and I'll just draw a few little Mycobacterium tuberculosis on this picture, they're all going to be activated to be releasing interleukin-12. And the way in which they're going to be activated to release interleukin-12 is actually through toll-like receptor 2. So remember, on the surface of the Mycobacterium tuberculosis cells, you have lipomannan, which I'll abbreviate to LM. Okay, and we went over that when we were discussing the structure of the cell wall of Mycobacterium tuberculosis bacterial cells. This can activate a pattern recognition receptor on the surface of immune cells, and the neutrophils will certainly have pattern recognition receptors on their surface, called toll-like receptor 2. And one of the things that toll-like receptor 2 activates is, remember, the release of pro-inflammatory molecules, and the two that we dwelled on were interleukin-1 and tumor necrosis factor alpha, and we saw how the release of these causes the inflammatory response, and of course the continued presence of the Mycobacterium tuberculosis and the continued activation of pattern recognition receptors on neutrophils, macrophages, dendritic cells, all of these cells of the immune system will continually cause them to produce interleukin-1 and TNF-alpha to maintain the inflammatory response. But another molecule that I didn't mention at the time that is induced to be produced by the activation of toll-like receptor 2 is interleukin-12. So these cells at the site of infection are going to be producing interleukin-12, all of them. The neutrophils, the macrophages, the dendritic cells, you name it, they're all producing interleukin-12. And of course, some of this interleukin-12 will be getting into the lymphatic vessels. And when it drains in the lymphatic vessels, where will it end up? Well, of course, it'll end up in the lymph node. So where the T-cell activation is occurring and the T-cell proliferation and the T-cell differentiation is occurring, you're going to get interleukin-12. And this interleukin-12, produced because of the activation of toll-like receptor 2 on those immune cells at the site of infection, and remember, the toll-like receptor 2 is detecting lipomannan, one of these pathogen-associated molecular patterns associated with Mycobacterium tuberculosis, you're going to get the interleukin-12 coming here, and that's going to be influencing the adaptive immune response that you're going to be initiating, and it's going to cause you to make T helper 1 type effector helper T cells, and these are the best type of helper T cells that you could produce for a tuberculosis infection, because these are going to go and help macrophages to, well, not just macrophages, they're going to help macrophages and dendritic cells, so not just macrophages. They're going to help majorly macrophages, though, to phagocytose and successfully destroy Mycobacterium tuberculosis cells. So, let's now go over T helper 1 cells. So I'm going to draw one of these T helper 1 cells in a little bit more detail here. So here is a big T helper 1 cell now, currently in the lymph node. So one thing that I want to mention is a molecule that these T helper 1 cells are going to express on their surface, which is really important. And this actually isn't specific to T helper 1 cells. All of the effector helper T cells produce this molecule and express it on the surface once they've differentiated into effector helper T cells. And this molecule is Cd40 Ligand. This is extremely important for the activation of B cells if we're talking about a full humoral adaptive immune response. In the case of Mycobacterium tuberculosis, we're not going to get that full humoral adaptive immune response, and the Cd40 ligand on these T helper 1 cells is going to instead be really important for helping macrophages. So this is a T helper 1 cell, and they've produced, all of them have produced loads of Cd40 ligand molecules which are now on their surface, and this allows them to actually do something to help out with killing the Mycobacterium tuberculosis. So what we now want to see then is we've produced all of these T helper 1 cells which have T cell receptors directed against fragments of antigens from Mycobacterium tuberculosis. How are these T helper 1 cells now going to actually help clear, or at least control, the Mycobacterium tuberculosis infection? Well, the T helper 1 cells are going to go into the bloodstream, and let me just remind you of how they actually get into the bloodstream. So remember, lymphocytes are continually circulating between the circulatory system and the lymphatic system. So T cells and B cells are not stationary in a single lymph node for their entire life, and these T helper 1 cells are not going to be stuck in that lymph node for their entire life. Instead, what happens is they gradually drain out of the lymph node, so they'll move into the medullary sinus of the lymph node here, they'll drain into the efferent lymphatic vessel, then they might end up draining into another lymph node, they'll make their way through that lymph node in the same way to its medullary sinus and into an even bigger lymphatic vessel, and eventually going through up the levels of the lymphatic system, they will eventually get into one of two major lymphatic vessels, which are the thoracic duct and the right lymphatic duct, which drain into the major central venous system. So let me just remind you of a bit of the anatomy here. So just drawing some pictures of the anatomy of the major superior venous system. This is representing the superior vena cava, which I'll abbreviate to SVC, which obviously drains into the right atrium of the heart. I'll remind you that the superior vena cava is made by the fusion of the two brachiocephalic veins. So here we have the left brachiocephalic vein, and here we have the right brachiocephalic vein. So these are the brachiocephalic veins, and each of the brachiocephalic veins are produced by the fusion of the internal jugular vein on each side with the subclavian vein on each side. So doing it on the left side here, we have the left internal jugular vein fusing with the left subclavian vein, and equivalently on the right side here is the right internal jugular vein fusing with the right subclavian vein. So just labeling this up, this is the subclavian vein which drains the upper limb, so subclavian vein, and here is the internal jugular vein which drains the head and neck. So internal jugular vein. Okay, right, so now let me show you where the major lymphatic vessels are going to drain into the central venous system. So you have two major lymphatic vessels. The bigger is the thoracic duct, and the thoracic duct, it lies in the midline in front of the vertebral bodies for most of the thoracic cavity. However, it moves over to the left-hand side as we approach the top. So here, this will show the thoracic duct. So it moves along the midline, and then as we approach the top, it moves over to the left-hand side. And as it ascends up, it's going to be draining lymphatic vessels from all over the body. Initially, when it's in the midline, it will be draining lymphatic vessels from both sides of the body, so left and right, and this green pen is really resisting being used. So I think I'll give up on using that pen. I'll colour them in yellow instead. And then once the thoracic duct moves over to the left-hand side, then you'll start just draining lymphatic vessels from the left-hand side, and instead you now have another major lymphatic vessel on the right-hand side which will drain the lymphatic vessels of the right side of the body, like so. Okay, so these two major lymphatic vessels then, this one is the thoracic duct which drains into the subclavian vein on the left-hand side just before it fuses with the left internal jugular vein to form the left brachiocephalic vein. So this is the thoracic duct, and then on the right hand side here we have the right lymphatic duct, which is another big lymphatic vessel, not quite as big as the thoracic duct, but still major. which will drain into the right subclavian vein, again just before it fuses with the right internal jugular vein to form the right brachiocephalic vein. So this is the right lymphatic duct. So why have I drawn this? Why have I shown you this? Well, it's just to remind you that the lymphatic system does eventually drain into the circulatory system, so these T helper 1 cells that are currently in the lymphatic system, they can get back into the circulatory system, and this is how they will get back into the circulatory system. So T helper 1 cells are going to drain through the lymphatic system back into the circulatory system. They'll eventually go into the venous side of the circulatory system, but of course the heart will move them all around the body, and of course we're talking about the lungs at the moment, so they'll go through the right atrium into the right ventricle, and then that will expel them into the pulmonary trunk, where they'll go into the pulmonary arteries, and therefore they'll go round the lungs. Now what's going to happen is they're going to be recruited to the site of infection. So where the acute inflammatory response is occurring, at the sites where the Mycobacterium tuberculosis is present, that's where T helper 1 cells will be recruited out of the blood vessels and into the tissue fluid. So T helper 1 cells are going to be recruited, just like we've discussed for neutrophils and macrophages, into the site of infection. So this is how we're going to get T helper 1 cells turning up at the site of infection. So now to add this onto the picture, you're going to actually get the T helper 1 cells appearing in here. So I'll just draw them like so, and we'll have them, well, I would have had them in green, but actually I know I said I wasn't going to use this pen again, but I'm just going to use it one last time. So there we go. Here are the T helper 1 cells now arriving at the affected area, and what we want to now understand is how are they actually going to help? How are they going to help us to contain the Mycobacterium tuberculosis bacterial cells? So, the way in which they help is they activate macrophages. The major cell type that they're going to actually help is the macrophages. They can do it to dendritic cells as well. All that you require actually to get the help is the presence of the MHC class II peptide complex, which the T helper 1 cells are directed against. But the major cell type that you've got a huge supply of coming into the area is macrophages, and therefore the major cell type that the T helper 1 cells are going to act through is the macrophage. So let me now discuss what the T helper 1 cells are going to do. So it's just like what we've discussed up here, only it's the other way around. Here we have the antigen presenting cell activating the T cell. Now we're going to have it the other way around. The T cell is going to activate the macrophage, but you're going to form a very similar junction to this. So you're going to form another immune synapse. So let me show this here. So we'll have the T helper 1 cell here. So here is its nucleus, and I'll just colour it in green again. Okay, so of course let's firstly just go over what's on the surface of the T helper 1 cell. So the T helper 1 cell, it has its specific T cell receptor, the T cell receptor which can recognise and bind to MHC class II protein complexes with the specific antigen fragments that these cells are armed against, presented in the peptide binding groove. Okay, so let me put this here. So here is the T cell receptor on the surface of the T helper 1 cell. So this is the T cell receptor, and I'll just colour this in blue. You also have the Cd4 molecule on the surface of the T helper 1 cell. And the other important molecule that you have on the surface of the T helper 1 cell is going to be the Cd40 Ligand molecule. But we'll talk about those in just a moment. So now what it's going to look for is it's going to look for macrophages that are evidently having problems with Mycobacterium tuberculosis currently present within their phagosomes. And these cells, generally, they might have managed to break down some of the antigens of the Mycobacterium tuberculosis. So you have to understand that there is a continuous struggle occurring between the phagocyte and the Mycobacterium tuberculosis cells that are inside the phagosome. The phagocyte is continuously trying to activate the mechanisms for killing and breaking down the Mycobacterium tuberculosis cells, and the Mycobacterium tuberculosis cells are continuously reducing that attempt, resisting that attempt. However, occasionally the phagocyte will succeed and break down little bits of the Mycobacterium tuberculosis cell, so it might break down a few of the antigen fragments of the Mycobacterium tuberculosis cell, and then it will end up presenting those fragments on MHC class II protein complexes on the surface of the phagocyte. So even though the phagocyte might not have succeeded in killing the Mycobacterium tuberculosis cell, it will end up getting fragments of the antigens broken down and presenting them on its MHC class II. And this is the way that the T helper 1 cells recognize which macrophages they should be activating. You see, these T helper 1 cells, they are targeted against a specific peptide fragment, and they are allowed to activate macrophages that are currently fighting the pathogen which has the antigen that has this specific peptide fragment on. They are not allowed to just go around activating whichever macrophages they like. That would be very dangerous, okay, because you might have macrophages that are doing things that you don't want to activate. So you only want to activate macrophages that are attacking a pathogen that this T helper 1 cell is actually targeted against. And this is the mechanism by which they're actually going to find which macrophages are attacking the pathogen that this T helper 1 cell is actually targeted against. So drawing this on then, this macrophage here, if it's got Mycobacterium tuberculosis cells inside its phagosome, and let's just draw this here, so let's say it's got absolutely loads of them in here, they've been breeding inside this phagosome, so you've got lots of Mycobacterium tuberculosis cells inside the phagosome. So this phagocyte is not doing very well, this macrophage is not doing very well. Despite that, it is coated in MHC class II protein complexes where little fragments of the antigens that it's successfully managed to break down are now presented. So here, let's say that this is the fragment of one of the antigens of Mycobacterium tuberculosis that is actually the fragment that this T cell receptor is targeted against. So, colouring this in, here this is the MHC class II protein complex on the surface of the macrophage. So I'll just label that up as such. So this is MHC class II. And then in the peptide binding groove of the MHC class II, we've got the peptide fragment there in green, and we've now got the T cell receptor binding to that peptide fragment MHC class II complex. Now, of course, that's not the only thing that will bind to the MHC class II. You'll also have the co-receptor on the surface of the T helper 1 cell binding as well, and of course that co-receptor is CD4 protein. So I'll colour the CD4 protein in red. So we've formed this nice immune synapse between the T helper 1 cell and the macrophage that is struggling to break down these Mycobacterium tuberculosis cells inside its phagosome. Now, the T helper 1 cell is going to deliver activation to this macrophage. It's going to deliver huge signals that are going to help promote the breakdown mechanisms for the Mycobacterium tuberculosis. So this is the role of the T helper 1 cells. I always give a comparison to a scene out of Harry Potter 4. So in Harry Potter 4, Harry goes to see Sirius, who is in a cave at the time, and Sirius starts telling Harry all sorts of stories about the time that Voldemort fell previously, once Harry was a baby and Voldemort lost all his powers and the Death Eaters were falling. He describes how Barty Crouch, the one in the Ministry of Magic, not the one that's the bad one, how Barty Crouch, who was very high up in the Ministry of Magic, rose to power whilst Voldemort was falling. And he describes to Harry how Barty Crouch fought fire with fire. He gave the auras new powers. He gave the auras the power to just kill Death Eaters and not have to capture them. So that's the comparison that I always use with what these T helper 1 cells are doing to macrophages. The T helper 1 cells are analogous to Barty Crouch, and the macrophages are analogous to the auras, and the Mycobacterium tuberculosis are analogous to the Death Eaters. So the T helper 1 cells are giving the macrophages the permission to use brutal mechanisms to destroy the pathogen that is within. They are giving them activation to use utterly brutal levels of these killing tactics to try and actually purge the Mycobacterium tuberculosis. So the macrophages have only been attempting to kill these Mycobacterium tuberculosis cells on a sort of half-hearted level so far, because they don't want to generate too many reactive oxygen species or too many reactive nitrogen species. So let me just discuss this in a little bit more detail. So you have to understand that when macrophages are attempting to kill what is inside their phagosome, you have the potential to cause a lot of tissue damage, because, for instance, when you generate reactive oxygen species and reactive nitrogen species, unfortunately they are not just going to be contained to the phagosome. Some of these molecules are going to diffuse out of the phagocyte and are going to reach your own cells and are going to cause big damage. So macrophages normally, they only attempt to produce reactive oxygen species and reactive nitrogen species at a sort of half-hearted level, because they don't want to produce too many reactive oxygen species and too many reactive nitrogen species that they'll then cause massive tissue damage. The T helper 1 cells, when they come and activate the macrophages, they are giving them permission to use go at it to the full level. They're giving them the permission to try as hard as they can with their killing mechanisms, produce as much reactive oxygen species and reactive nitrogen species as you like. Fight fire with fire and don't worry about the consequences of it. That's what the T helper 1 cells are doing with the macrophages. They are activating the killing mechanisms to try and get the macrophages to kill these mycobacterial tuberculosis cells. So, let's continue on with the story now. That's the motivation of where this is going. So, how is it actually going to occur? Well, the other part of this interface, this immune synapse, is that the T helper 1 cells have on their surface Cd40 Ligand molecules, which we've discussed all effector helper T cells will put on their surface and start expressing when they differentiate into effector helper T cells, when they became effector helper T cells. And this is now going to activate a molecule on the surface of the macrophages known as Cd40. So here is Cd40 on the surface of the macrophages, and that's going to activate killing mechanisms inside the macrophages. There is another thing that the T helper 1 cells do to activate the macrophages, which is that they release a molecule known as Interferon Gamma, which for short is called IFN Gamma. So this is Interferon Gamma in full. So I for inter, F for fur, and then N for on. So Interferon Gamma here. So they release the Interferon Gamma, and Interferon Gamma is going to act on Interferon Gamma receptors on the surface of the macrophages. So let's say this little protein here, this can represent Interferon Gamma receptor, which I'll colour in blue here. And that's also going to start intracellular signalling cascades that are going to lead to a redoubling in the effort to actually kill these Mycobacterium tuberculosis cells. Okay, so overall the T helper 1 cells then, they can go up to macrophages, and if the macrophage has on its surface MHC class II proteins with the correct peptide fragment that this T helper 1 cell is targeted against, the T helper 1 cell will form an immune synapse like so, and the Cd40 ligand proteins on the surface of the T helper 1 cell can then activate Cd40 proteins on the surface of the macrophage, as well as interferon gamma that the T helper 1 cell is releasing, activating interferon gamma receptors on the surface of the macrophage, and these two mechanisms will activate killing mechanisms inside the macrophage, and this hopefully will finally lead to some destruction of these Mycobacterium tuberculosis cells inside the phagosome, even if there is a cost to the surrounding tissue because this macrophage starts leaking a huge number of reactive oxygen species and reactive nitrogen species. Okay, so at this stage I want to also remind you of the fact that you will have produced T helper 1 cells of different clones. Remember, the Mycobacterium tuberculosis cells, they have a huge number of different antigens. Each antigen has a huge number of different peptide fragments, so you can get lots of different T helper 1 mediated adaptive immune responses initiated, where you have different T helper 1 cells that have different T cell receptors directed against different peptide fragments from the Mycobacterium tuberculosis cells. You won't just be doing this for one peptide fragment, you'll be doing it for loads of peptide fragments. So you'll get loads of T helper 1 cells being produced, and these are going to assist the macrophages. Now, we're going to have a break here, and in the next video what I will go on to is the massive topic of granuloma formation. And I'm afraid this is the point where mechanisms start to become more fuzzy. We don't know exactly how granulomas form. So what I'm just going to have to do is go to description. I'm going to describe what is going to happen and how this actually contributes to a latent tuberculosis infection. I'll also talk about what happens in immunosuppressed people, because all of what we've just been discussing about this adaptive immune response, this is only going to occur if you are immunocompetent, and we'll also discuss secondary tuberculosis in the next video. So we'll have a break here.